Morning, students. In previous lecture, we were discussed about the structure of muscle or the structure of all type of muscles like uh, heart muscles, skeletal muscles, and non-striated muscles, or we can say visceral muscles. Today, we will discuss about molecular structure of striated muscles, or the molecular structure of a skeletal muscle. I am Dr. Ashok Kumar Siroya, Associate Professor, Department of Zoology, SPC, GC, Ajmer. The molecular structure of striated muscles. We can study this as a structure of sarcomere, because sarcomere is a unit of the striated muscles. In a muscle fiber, between two Z lines, the specific area of two Z line is known as sarcomere. So sarcomere is a unit of muscle. Here we can see the Z line. In between these Z lines, there are many different type of proteins lies here. These are actin, myosin, tropomyosin, troponin, and other structural proteins. Sarcomere, area of sarcomere can be divided the middle M line and area beside M line is known as H zone, and the anisotropic area, which is lies in between Z lines, is known as A band, and between A band of two sarcomere, there is an area of anisotropic. This is called I band. I band, we can say the area beside the Z line in anterior and posterior part is known as I band. There are three type of proteins found in a sarcomere to facilitate the contraction process. These three proteins are force generating proteins, regulatory proteins, and structural proteins. First one, the force generating protein, are myosins and actins. Second, tropomyosins, and troponin. These are the regulatory proteins. Third type of proteins are the structural protein, which lies on a sarcomere, consists some structural part of that sarcomere. The first one is alpha actinin, second one is M disc protein, and third one is C protein. Now we will discuss in detail about force generating protein. The force generating proteins are also known as contractile protein, or we can say these are the main proteins involved in contractile process, contraction procedure. These are thick protein filaments made up of myosin and the thin filaments made up of actin. The first one is myosin, has a molecular weight of about 450,000 unit. It is tit ball shaped and composed of two globular head and a long tail. The heads form cross bridges with actin, and a myosin molecule can be broken into head and a long tail fragments by the enzyme trypsin. The head fragment is globular, about 150 to 250 degree angstrom long and 40 degree angstrom in diameter, and molecular weight of about 350,000 unit, called heavy myromycin. It has two active sites. One binds with actin molecule and other is catalytic site where hydrolysis of ATP occurs. The head fragment is the main part of the myosin. We can say it HMM, called heavy myromycin. And the tail fragment is known as light myromycins, or LMM. It is about 1000 angstrom long and 15 to 20 angstrom in diameter, having the molecular weight of about 150,000 or 150,000 unit. Here is the diagram of actin. These are the actin filament and these are the myosin filament. In between myosin, or in between actin, there is a gap. We can say it breeze free region, and myosin is continuous between the two actin filaments. This is myosin filament. It has head part. There are two head part and the tail part. One is attached to actin and one is attached to ATP to generate energy. And these are the globular part arranged to make filamentous part of actin. Actin is coiled filament of globule protein. These are the force generating protein. Second one is actin. We have discussed about actin. Second one is actin has a molecular weight of about 70,000 units. It occurs in two forms, a globular G form or filamentous or F form. Actin attain a globular form in the presence of salts. In the presence of KCl and ATP, it polymerizes to long fiber or F-actins, which is made up of two helical chains twisted around each other. These are the globular form and these are the filamentous form. In presence of KCl and ATP, they make a string and the string coiled each other and form the fibular form. It is known as polymerization of actin. In next lecture, we will discuss about other type of proteins which are regulatory proteins and structural proteins. Thank you.